The title of my talk is Synuclein gamma, SNCG, and the Chaperoning of the cancer. I will have my talk briefly in three parts. In the first part, I will give you a brief introduction on the gene we cloned in 1997, 12 years ago. At that time, the gene was named as breast cancer specific gene 1, SNCG1, and later the gene was renamed as Synuclein gamma. In the second part, I'll give you a highlight, basically a summary and some of the key data showing this protein can chaperone several major pathways involved tumor cell survival and growth. Basically, steroid receptors, estrogen receptor, AKT mTOR pathways, and also in mitotic checkpoint function. In the last part, I will discuss with you some clinical application of using this protein as a biomarker to predict patient response to certain chemotherapy drugs, primarily Taxol-based therapy. In that regard, we just finished a pilot neoadjuvant clinical trial. This in collaboration with Xinhua Hospital in Shanghai. The data were promising, so I'm going to highlight some of the key points in that trial. Synuclein gamma were first identified and cloned by differential cDNA sequence because it was identified in the cDNA library from breast cancer, and the expression cannot be detected in any other normal tissues except brain. At that time, we gave a name SNCG1. When we put the sequence in the database, it turns out this gene shares no sequence homology with any other known growth factors or oncogenes, but rather have extensive sequence identity with a protein in neurological area called synuclein. At that time, synuclein alpha and beta already cloned. Now, our protein have over 55% sequence identity with alpha and beta, so it was renamed as Synuclein gamma. So right now, synuclein family has three members, alpha, beta, and gamma. In contrast with alpha, beta, which play a major role in neurological disease, like Parkinson's disease, Alzheimer's disease, gamma is not well studied in the neuron cells, but extensively studied in the malignant tumor cells. Since the first discovery, we first demonstrate expression of Synuclein gamma in breast cancer follows a stage-specific pattern. In other words, in a normal mammary gland, you cannot detectable. In the benign hyperplasia, it's a very low percentage expression, about 3 or 5%. In the early stage, stage 1 or 2 breast cancer, they're about 25%. However, in the advanced late stage, stage 4, which is infiltrating breast cancer, is over 79%. And this pattern has been almost demonstrated in virtually every solid tumors has been so far studied. There are 12 tumors has been studied, which including liver, the brain tumor, neuroblastoma, pancreatic, of course breast cancer, prostate cancer, cervical cancers. So we are interested how this pattern was really turns on. So when we look in the normal epithelial cells, we found out in the promoter region of this gene, particularly exon 1 and CpG island, is hypermethylated. Now, if you're treating normal cells with demethylating agent, you demethylate in the CpG island and turn the gene on. And if you look in the late stage malignant cancer, in most stage, this CpG island was demethylated. So it's an epigenetic control. The demethylation would turn the gene on in the late stage breast cancer and other types of cancer.